We'll talk about the cell membrane structure. So if we look at the cell membrane, why is it over there? It's very important. It plays a role in separating the ECF from the ICF. Extracellular fluid has different composition from intracellular fluid, and this composition of the ICF must be maintained in order to allow the organelles in the cell to function properly. So you have your nuclei, your mitochondria, your ER, and your Golgi apparatus. All those things require a certain environment in a way. So to maintain this, we need the border, something that will help in regulating the environment for these organelles to function and for life to take place. Now, so the structure of the cell membrane was given by Singer and Nicholson in 1972. They got the Nobel Prize for that. They coined the structure as they described it as a fluid mosaic model. So what does that mean? Fluid mosaic model. Mosaic refers to, if you remember, those tiles in Morocco or in the Middle East where they make different color clay tiles and they break them into little pieces, and when they break them into little pieces, they get these individual little building blocks of different shapes. So sometimes you'll get the rectangle shape, you can cut it into triangle shapes like this or diagonal shapes. So, and then they fix them into little shapes over here next to each other. So this is a mosaic pattern that you see. They make good tiles over there, and so they arrange them in certain ways, and that's how you get the mosaic. You get these designs over there based on the different shapes that you cut from here. Now, fluid meaning that it is able to move. These cells are more like imagine you take a drone camera and you go above an area like in a market or in a big bazaar where a lot of people are just walking around minding their business in different directions. So that's what your different components of the cell membrane are doing. They exist in a sort of mosaic pattern, meaning there are different molecules and like proteins, phospholipids, carbohydrates, all located over there in your cell, and they are able to move in two-dimensional space, two-dimensional plane, like how you are able to walk in a bazaar with people all around you, and so there are the stalls over here and there are the different people, and they're able to move around in different directions, but nobody can climb on top of each other. So that's why we say it's two-dimensional motion, 2D motion. That's why it's fluid, because there's two-dimensional motion. There is an example of where three-dimensional motion takes place, and we'll talk about that later. It's when one of these phospholipids called phosphoserine moves from one side of the membrane to the other side during times when the cell wants to undergo apoptosis and cell death. Anyway, we'll just go into that later. Now let's just discuss about the 2D motion. So it's all fluid-like in nature. The cell membrane is fluid, it's able to move, and yet it is made of different components that somehow organize themselves into a bilipid layer, which is around 5 to 10 nanometers thick. So you can see this picture over here I've drawn, and you could see that there are these little phospholipids over here. These phospholipids you could see like this, which I'm shading over here. This is a phospholipid head over here, and it is hydrophilic, and you have two tails over here. Now see, you have lipids. The phospholipids are quite abundant in terms of numbers. You have cholesterol, that's also part of the lipids over here. So you have phospholipids, you have cholesterol, you have proteins, which I've drawn in green over here. So you've got these integral proteins, which are really lodged inside the cell membrane, and you have these peripheral proteins over here, which are sort of connected over there loosely to the cell membrane by covalent bonds. The integral proteins are bound by van der Waals forces. Okay, van der Waals forces, van der Waals forces. Alright, so you have your van der Waals forces and hydrophobic interactions. for the integral protein. For the peripheral protein, you have got your this one. They are weakly bound by covalent bonds with your integral protein. So they are stuck to the integral proteins also, and by your electrostatic interactions and hydrogen bonds. So you have your electrostatic interactions and hydrogen bonds that help with the peripheral proteins. There are some other proteins called lipid anchored proteins. These proteins are bound to a phospholipid called not a phospholipid, an important glycolipid called glycosyl phosphatidyl inositol. Lipid anchored proteins are bound to the cell membrane via glycosyl phosphatidyl inositol. Very important to remember. Okay, so these are the proteins over here, and you have lipids and you have some carbohydrates and sugars over there which are located also. We'll talk about that in some time. Now, predominantly in terms of numbers, we have phospholipids as the major component. Now, your cell membrane is asymmetric. What does that mean? Well, you could see that first of all, it's a bilipid layer. You have two layers of your like a sandwich. It looks like a sandwich. So here you have these phospholipids which are arranged end to end over here, and you could see that they have these head regions over here, and they have two tails coming from the phospholipids. Why is this? Because most of these components, not just phospholipids, even your proteins, they have got a part that likes to face water, because look, this is the cell membrane and there's water on two sides. There's extracellular fluid outside and there's intracellular fluid. So what happens is these molecules, some of them, a part of them likes to mix with water and a part of them does not like to mix with water. So you have these hydrophobic regions or water fearing regions which don't like to face the water, and you have hydrophilic regions or parts that love to face the water. So the hydrophobic regions organize themselves, in a way that they don't mix with water. So you see these tails over here, these fatty acid tails, they hate water. So they stay in the middle of the sandwich layer. See, it looks like a sandwich. And the hydrophobic regions, the hydrophilic regions, the ones that like the water, these head ends, they will face the water. Okay. Same thing with proteins. There is a portion of the protein structure which faces the water, and that tends to be the hydrophilic or water loving area, and the part that is lodged in the membrane, in the middle of the membrane, not facing water, is hydrophobic part. Okay, so all of these things have got hydrophilic and hydrophobic regions over here. Right, and they all are, some of them, most of them are able to move around in a fluid mosaic model, but some of them are anchored to cytoskeletal proteins, and that doesn't allow them to move too much. Right? And other than that, you should also be aware that, so what is asymmetric structure? So look, for example, epithelium, the epithelial surface, this is a cell, epithelial cell, can you see? Let's say it's in your kidney, for example. So let's just say that there is an epithelial cell in your proximal convoluted tubule, PCT, proximal convoluted tubule of your nephron. So there's a part that faces the lumen of the nephron. So here's your nephron, and this is the lumen. Here's the glomerulus over here, which I draw in red, and there is a lumen. This lumen carries the filtrate, the glomerular filtrate. And now you have cells over here. Now if you see, there is a part of the cell which is facing the lumen, the luminal side. We call this the apical side. And you have something called the basolateral side. Basolateral. Now what happens is you have to reabsorb sometimes sugars, salts from one side to the other. So that means there are different transporters and carriers that are located on different surfaces over here. That's what we mean by asymmetric. So there are regions or borders on the epithelial membrane which isolate proteins on one side from another side so that they don't cross to the other side. Think of it like a belt around the stomach, and something cannot cross that to the other side. It's not possible. So this contributes to asymmetry of the membrane. And if you look at the membrane over here, it is fluid in nature also in terms of it acts. There are states called gel state and sol state. So if we look at the gel state, this is more like a solid state which happens at lower temperatures. And as you keep increasing the temperature of the membrane, it becomes more fluid and more effective in moving stuff around, and that becomes a sol state. So there is a temperature at which the cell fluidity changes from a gel state to a sol state, and this is known as transition temperature. Okay, anyway, here's a nice picture of all these cells in the fluid mosaic model over here. And you also get carbohydrates. You can see I've drawn in red over here in this picture, and they are sometimes lodged on different proteins or different lipids over here. And the carbohydrates are always on the outer leaflet of the membrane. So like I told you, this membrane is organized like a sandwich, and we have an outer leaflet that is facing the ECF and an inner leaflet that is facing the ICF, intracellular fluid. And the carbohydrates are always on the outer leaflet. They face the outside of the cell, not the inside that much. Now if we look at the number of molecules over here, protein to lipid, of course the ratio of lipid in terms of numbers will be high. So the ratio of protein to lipid is 1:50. But in terms of mass, depending on the cell, most of the time protein to lipid ratio by mass is 1:1 ratio. Now it is different in some different cells. For example, where do you need more protein? Where the cell has more activity. Proteins are the building blocks of life, so sometimes you need proteins to make transporters, to make enzymes, to make important complexes over there. So for that, you have some cells like the inner mitochondrial membrane, which is important for electron transport chain to make, to need a lot more protein. So the ratio is 3.2:1, more protein than lipid over there. Which is much more than in the sarcoplasmic reticulum, which, as you know, has a lot of calcium pumps. Sarcoplasmic endoplasmic reticulum, ATPase pumps. And this is going to pump a lot of calcium and store it in the sarcoplasmic reticulum, so you have a lot of protein over there. Then you have your outer mitochondrial membrane. This is going to have almost 1.1:1 protein to lipid ratio. It's also almost the same in the RBC, around 1:1 ratio almost. The lowest ratio of protein to lipid is myelin, because what is myelin? It's the insulator for the neuron. It doesn't have to do too much in terms of I mean, it does have its role to play, but I'm saying that it's more like an insulator for your neurons.